Okay, so let's talk about the immunity. What type of defense mechanisms are there in place to be able to resist pathogens? So there are two different types, innate and adaptive immunity. Innate immunity is that very broad, very rapid response. And when I say broad, it recognizes very non-specific type of pathogens because we have different barriers in place, such as skin and mucus and secretions. And if the pathogens do manage to get through that barrier defense, then internal defenses are going to be taking place. So it means phagocytic cells, natural killer cells are going to play the role. Gene expression is going to be changed. Antimicrobial proteins produced to fight the invading pathogens. Then we also have inflammation, which is an inflammatory response, and complement system with interferons that play a huge role in cell signaling. Adaptive immunity, on the other hand, is going to recognize traits of a specific, very specific pathogen because there are going to be receptors that are going to be binding to antigens, and it has to be this perfect match. Therefore, the system is much slower. It can actually take days, if not weeks, for the system to be functioning. There are two types of responses in adaptive immunity, humoral and cell-mediated. So humoral one is going to be using B cells, and there we also see antibodies that are going to be capturing pathogens. And then in cell-mediated response, T cells are going to be playing a huge, huge role. So this picture summarizes the cells of the immune system. So you can see how they all related and that they all come from stem cells. And the ones that I've circled in blue, these are cells that play the role in innate immunity. So notice natural killer cell. These are granulocytes. And then we have dendritic cells and macrophages. So these can also become what we call antigen-presenting cells because they're able to phagocytose microorganisms. And granulocytes, notice, contain different types of granules that are going to be released once they get the signal. So that way they can elicit a response such as inflammation. So if the pathogen gets through the physical barrier, macrophages are going to be those first responders. They're going to arrive to the scene and try to phagocytose the bacterium if that's what entered the tissue. So macrophages are going to process, they're going to break down bacterium, and they're going to take these little remnants of bacteria that we call antigen, could also be virus particles, could any sort of remnant from a pathogen is considered to be an antigen, that non-self structure. So they're going to present antigens by embedding them in special molecules that we call major histocompatibility complexes, and they're going to be presenting these fragments, sort of sticking them out on the surface of the cell so that other immune cells can recognize them. They will also produce cytokines, which are chemical messengers, and they're going to be calling for help, calling neutrophils, dendritic cells, granulated cells. So they're all going to be drawn in from the bloodstream, and they're going to move by chemotaxis because they're responding to those signals. And then notice the basophil cells and mast cells, they're going to be releasing their granules. And for example, mast cells release histamine, and histamine causes blood vessels to dilate. So, and then neutrophils will arrive to the scene and try to break down the pathogens that are there or engulf them. And then macrophages can present the antigens, and it's not just macrophages. We can also have neutrophils and dendritic cells that can be antigen presenting cells, so they are going to activate adaptive immune response when innate is not enough. So natural killer cells, as I mentioned before, are going to be activated by macrophages, but they also have the ability to circulate and identify tumor cells, and then they kill them before a person realizes that those cells are there. So let's take a look at what happens when the pathogen binds to the receptor of the cell. So there's this toll-like receptor that a pathogen piece, or this tiny little pathogenic molecule that binds to the receptor, this receptor will be activated and it will initiate signal transduction pathways. So here we see phosphorylation cascade, and then protein downstream is activated, and it serves as a transcription factor because it's binding to the DNA in the nucleus at the promoter region and initiates transcription. So transcription is going to produce or result in mRNA. That means the gene is expressed, and then mRNA will be translated into the protein. So here we have produced a defensive protein. This could be a lysozyme that specifically targets peptidoglycan cell wall in bacteria, or it could be defensin, which is another antimicrobial protein that targets the cell membrane by embedding itself there and forming holes in the membrane. So you can see how a little tiny piece of pathogenic fragment is initiating the signal transduction pathways, essentially changing the gene expression. So another factor that can happen is basically the release of cytokines. So cytokine is a chemical messenger, and we have over 40 different types of cytokines, and give you one example, which is interferon. So when interferon is released by infected cells, notice there's exocytosis of this tiny little vesicle, infected cell, infected by a virus. This signaling molecule is going to signal neighboring uninfected cells to be on alert and be ready to destroy, let's say, viral RNA, because the virus is in the vicinity, and potentially change the protein synthesis. If the nearby cells are already infected, that signal may trigger apoptosis, so that cell is going to die. And then this particular signaling molecule can also activate other immune cells, so we can move on and get help from other immune cells out there. So inflammation is another response to infection or injury. And it is a very coordinated response. And basically what we see here is the redness, swelling, heat, and pain in the damaged site, because you can see here we have a splinter that broke through the epidermis into deeper tissues and deposited pathogens. So those would be bacteria. Macrophages arrive to the scene because they sense bacteria and they start phagocytosis. They release signaling molecules, and we discussed that. Those would be cytokines. Mast cells are also going to release their molecules, and those would be histamines. And histamines are going to cause the blood vessels to open up, and then there will be more blood flow, and also blood flow to that particular area, because now those signaling molecules are inviting neutrophils to the scene, and you can see how they're squeezing through the tiny channels in the blood vessel, and they are all engulfing the bacteria. They're breaking it down. So you would have that swelling going on here, the redness, and then high temperature potentially. And then the healing begins. Notice how the blood vessels go back to their normal state. So it means the infection has been cleared. Now, sometimes we say that inflammation is just inappropriately strong. Sometimes your body reacts to certain allergens extremely in a very, very strong fashion. For example, you are allergic to pollen grain, and then the next thing you know, you've got hives and runny nose and itchy eyes because your mast cells are releasing histamine, so they're basically causing inflammation throughout the body. Sometimes the reaction is so strong that a person can go into anaphylactic shock, so that could be a life-threatening condition if not taken care of immediately. Autoimmune disease is another problem where your immune system cells attack their own tissue, so they start to break it down. An example would be rheumatoid arthritis. So this is where your joints are going to be compromised. Sepsis happens, or septic shock happens, because of bacterial infections, and that would be also a very widespread inflammation. It's a medical emergency. because of the toxins that are released by bacteria, you've got degranulation of cells and the blood pressure drops significantly, and that becomes a life-threatening situation. Natural killer cells are going to recognize and attack cells that are missing or have a deformed major histocompatibility complex class 1. So what this means is MHC1. These are self-markers. These are glycoproteins that are embedded in cells, and that's how cells recognize themselves, because you would have a very specific pattern for that particular protein. And normal, all healthy cells would have those complexes, but sometimes if the cell is cancerous, they may not be expressing that protein, so that protein is missing. So natural killer cells circulate and look for those missing complexes. And if they do detect them, because they make direct contact, as you can see in this picture here, the natural killer cells are going to release enzymes, and they will cause apoptosis to that abnormal cell. Complement system proteins are a bunch of proteins that are present in your blood, and they are ready for immediate action as soon as there's a pathogen that they encounter, as soon as they encounter the pathogen in the system. So they work in a cascade type of fashion. So one protein activates another one, and you can see right here, and this one activates another one. And then in the end, there's going to be an effect. And in this case, these proteins came together and they formed a pore in microbial cell membrane. So what's going to happen to that cell is going to lyse. It means this is it. Traffic is no longer regulated, homeostasis is compromised, the cell is going to die. So the reason we call these proteins complement system part of proteins is because they sort of work together with antibodies. They complement these antibodies. When you have an invading cell, so this would be a pathogenic cell with the antigen, antibodies that you already have are going to bind to the antigens, and then complementary proteins will bind to the antibodies. So they're going to coat them. This coating also helps or enhances the engulfment of that pathogen. So let's say there is a phagocytic cell nearby, and it would have to engulf this pathogen to get rid of it. But now, because it's coated with the proteins, that engulfment is going to be enhanced. So we call that process opsonization.